So with this, I would like to introduce the next speaker, who have introduced herself already. This is Manasi Nandi. She's a reader in integrative pharmacology at King's College London. She started her academic career specializing in vivo pharmacology and now transitioned into data science. And she will talk about how to make better use of routine physiological signals and using math to improve the sensitivity of detecting drug or disease-induced changes.
Thank you very much for the invitation. It's a real pleasure to be here. So yeah, my name's Manasi Nandi, and I'm going to tell you a little bit about a technology that I've been co-developing with a mathematician, Philip Aston, at the University of Surrey. And I'm going to be talking about this image on the top left-hand of the screen. And this is actually the data from my arterial pulse waveform, which has been replotted and revisualized. And this image is giving us an indicator of the morphology changes of my pulse wave and the variability changes of my pulse wave. So I hope to show you that over the next 20 minutes that by including this extra information, you can potentially increase the sensitivity and specificity of looking at drug or disease-induced changes. So the idea of looking at morphology is not new, and people who already collect physiological waveforms will probably see it in their data anyway. This is from 1874, and it shows a pregnant woman at different stages of pregnancy, during labour and postpartum. And using this non-invasive device, you can see that not only can you measure the number of beats, which is usually what we measure, but actually you can see there's great variety in the shape of the waveform. And that must relate to changes in her cardiovascular system adapting through these changes during pregnancy and postpartum. Now, in 1874, we didn't have the digital data. Now we do have the digital data. And unlike this kind of image on the left, what we now collect is tons of numbers. So typically, in the preclinical setting, you're collecting between 500 to 1,000 data points per second. So here, what you can see on the right-hand side is just 1 second's worth of data captured from a mouse radiotelemetry. So it's too many numbers. Too much for us to handle using conventional methods. And so what we ask the machines to do is hide those numbers in the background and just show us the waveforms and give us some extracted metrics. So we get the extracted averages of the maximum, the minimum, the amplitudes, and the intervals. But what we're doing is we're throwing away most of the data that's available. So if you think of the waveforms on the left as the true waveforms where every point is actually as important as the next point, instead what we're doing is the thing in the middle, which is where we're saying these particular points are the points that are most important. And they absolutely are. We do use them. They're very useful biomarkers. But the problem is, A, you're throwing away most of the data, and B, it's not always perfect. So if you have movement, which animals and humans do, you're going to get baseline wonder. And if you get baseline wonder, you're going to get compromised detection of these single points. So you introduce error. So what I hope to show you is try and think of a waveform as where each point is as important as the next, and the relationship between them is the thing that we perhaps need to look at. So in addition to morphology, we also know that variability is also important. So is your heart or are you breathing in a kind of very rhythmic, periodic way? That tends to not be a good thing. Or is it more chaotic? And so that variability is also very, very important. But again, we don't really routinely measure it in a consistent way, either in the clinical setting or the preclinical setting. And the main reason is this issue of baseline wonder. And we're not really ever going to get around the issue of baseline wonder. Animals and humans will always move around. So this is a mouse blood pressure data. It could easily be a human. And you can see that the algorithm has put a dot on the points that it thinks are beats. And you can see around 3.5 seconds, it skipped a couple of beats. Now, if it systematically keeps doing that, you're going to get a systematic underestimation of rate, overestimation of variability. So can we look at this data in a different way and try and avoid these issues? And so this method is a very long name called Symmetric Projection Attractor Reconstruction, which I'm going to call SPAR for short. And as I said, it's developed by a mathematician, a dynamical systems mathematician, and myself, who's a sort of biomedical pharmacology background. And so I'm going to show you the maths first without any equations. And if you understand the maths, then hopefully you will be able to follow the kind of application of the method. So the thing that I need you to remember is that the waveform is made up of individual numbers. So there's a numerical value for every point on the waveform. And at the moment, it's plotted as pressure against time, but we're going to try and replot it in a different way. So this is my blood pressure data. We take a random point, we call it X, and then 2 further points, Y and Z. And we can read those numbers off the Y-axis. Now, these points are separated by a fixed amount of time. So if my heartbeat or pulse wave is 1 second long, then it's 1/3 of a second, the gap between these points. So they're equidistant. And it doesn't matter where we start, but I happen to have started here where X is at the bottom. So instead of plotting the data as pressure against time, we can plot the values against each other, X against Y against Z. So pressure against pressure against pressure. And when you do that here, you get a point in a cube. So that's the relationship of the 3 points in 3-dimensional space. And then the points just dance through the wave, and they touch every single number, and those numbers are then replotted in the cube. And every time those 3 points complete one pulse wave, we get a loop in the cube. So just showing you that, looking at that sort of big data in the background. Here we've got that big data, most of which we ignore. These are our first 3 points. That's a point in a cube. Then it shifts to the next 3 points, and that point will move, and so on and so forth, until we complete one cycle, and that generates a loop, and then here we've got about 8 cycles. So that's phase 1. Phase 2 is then to look at it and say, well, it's an absolute mess, and how can we possibly extract anything from it? This was just me under resting conditions, and it's really noisy. So if it's me under resting conditions, it's going to be impossible to kind of extract anything useful. But what the mathematician did, and this is something that I wouldn't do, is just look at the data from a different angle. And I think that's the real benefit of this sort of true interdisciplinary working, that you really look at things in totally different ways. And what you can see that by rotating that cube and looking at it head on, all of that noisy stuff, we haven't deleted it. It's just not visible anymore. It's going in and out of the screen. And now you're left with something with structure and form and shape. Now, the take-home message here is that that is just relating to shape. So if there's a small change in the shape of the waveform, so you get a tiny little change there, that might be difficult to see by eye, but it will be amplified as a bigger change here. And so what you're doing is taking 3D image, which is noisy, and you're looking at it effectively in 2D. The 3rd dimension is going in and out of the screen. So why would we do that, and what's it actually achieving? Well, if we go back to our old friend, baseline wonder, here again we've got a perfectly healthy mouse, happened to be wandering around, spike in blood pressure, and then it bobbles along. If you plot that in 3D, every wave is different to the next, and it's making this really noisy 3D construct. But if you rotate it and look head on, most of that noise goes. So that rotation is effectively ignoring the movement in the original y-axis. It's factoring out baseline wonder. As a result, the units of that image are not millimeters of mercury, they're just shape units. It's just a shape of a wave. It doesn't care what the wave is. So now we can look at morphology and we can quantify it, and then we add color, and that tells us about variability. So if this was an engineering data, if it was from an engine and it was perfectly periodic, then this would be a single red triangle. So the blue here represents the variability in this mouse from beat to beat. And so this is what it looks like over time. Again, this is just me under resting conditions, and you can see it's just amplifying what's going on in the signal. It's easier for us to see because the waves are stacked on top of each other. And so you can imagine if this is me under resting conditions, if you then gave me a pharmacological insult, or if I was developing a disease, in theory, that should make quite marked changes on this. And so in addition to the image, which probably makes zero sense to anyone in the room, but I can kind of interpret them now, we can actually extract metrics. So we can look at the length of the arm, the rotation, the densities. There's so many different metrics that you can get. And some of them are going to be more physiologically relevant than others. So we need to look systematically in data to work out which ones mean what things. And ultimately, this is saying use all of the data, get this extra information about morphology and variability, add it to the conventional biomarkers that you're already using. You've therefore used all of the data, and can you more sensitively detect change? So obviously this is a respiratory conference and I've just talked about blood pressure for most of my talk, but as I said, it doesn't care about the y-axis, so the method doesn't care that that was a pulse wave or a respiratory flow wave or an ECG, it's just looking for a cyclic signal. So I'll now focus and tell you a little bit about the application of the method to respiratory function. So the data I'm going to show you is two sets of data, a non-clinical data study from Charles River Labs headed by Mary McElroy looking in a lung fibrosis model with bleomycin in rats measured using head-out plethysmography. And the second is looking at human data, patient data from St Thomas's Hospital collected by Rebecca de Cruz, and that's looking at COPD patients and working out whether they've got an ensuing exacerbation. So we take the data, and the beauty of this data is it was very well curated and very well annotated. We then do the minimal amount of cleaning. As I said, we don't have to kind of select less noisy areas because baseline wonder is not a problem, but we do take out electrical noise or things like that that are non-physiological. And then we do the conventional analysis that everybody does, and then we make an attractor, and then we do the attractor analysis. And then the question is, does having this additional information improve the classification accuracy between two groups? So first case study is a bleomycin lung fibrosis model. As I said, this was work done by Charles River Labs, and they gave us the data, and it was the respiratory flow data. And in the circles, in the green circles, you've got the head-out plethysmography measures. So on these 5 days, the animals were monitored. In the red arrows, you've got the bleomycin dosing, and in the black, you've got the nintedanib dosing. So we basically got 3 groups: vehicle control, bleomycin alone, and then bleomycin with the standard of care. So just to confirm that the model worked, they sent us their histology data, and you can see that bleomycin has the sort of most marked effect on fibrosis and inflammation. It's attenuated with the standard of care, and it's absent in control. So we know that the animals had the phenotype that was expected. And again, looking at their data of conventional metrics, in red you've got bleomycin, in blue you've got nintedanib, standard of care, and in black you've got vehicle. And you can see that the sort of blue falls intermediate, so you're seeing the greatest effect with the bleomycin, it's intermediate with the nintedanib. And you can also see that the sort of biggest changes are happening in the first few weeks, and then they sort of die down and the error bars start to overlap a lot more towards the end. So there are differences, they're statistically significant, but you would have some difficulty in classification because there's quite a lot of variability. So can we add more information by looking at this data in a bit more detail? So looking at the attractors, and these are just 3 animals that I pulled out, so these are sort of semi-representative, but it was sort of random. So in the top you've got vehicle, and you can see that largely they form this kind of helicopter structure, a 3-armed structure. With the bleomycin, you can see it starts with the helicopter structure because it's a control, and then it goes kind of round and fuzzy and diffuse in the first few weeks, probably when there's edema and inflammation, and then it becomes a little bit kind of more round, but then with some hot spots as the fibrosis develops. And then in the final one, the nintedanib, you can see it's sort of intermediate. It's got a mixture of the helicopters and a mixture of the circles and a little bit of fuzziness that then dies down. But more or less by day 21, day 26, it's looking a lot more like the vehicle. So then we can quantify these, and one of the metrics we use is the sort of opening of the attractor. Is it sort of a helicopter shape? Is it closed in the middle, or has it got a hole in the middle? And looking at the waveform, and this kind of sort of talks to the last speaker, when you normally breathe, you're 1/3, 2/3. You breathe in fast, you breathe out slow. And that generates this sort of triangular shape. With bleomycin, it becomes more sinusoidal, and that generates a rounder attractor. And then with nintedanib, it's sort of halfway between. And what you can see here is respiratory rate in the graphs on the left, and you can see there are differences, but using ROC-AUC, which is a sort of more granular form of looking at sensitive and specific differences, you can see it's not brilliant at detecting the differences, so respiratory rate is not the best classifier in this situation. If you look at the graph on the right, you can see that the opening of the attractor improves that classification accuracy, not by loads, but it's certainly a lot better. And you can see that the bleomycin is markedly worse than the nintedanib group, and the nintedanib is similar to the control. So this first part of the data what we think is if we add SPA to the conventional metrics, we can increase the sensitivity and specificity of quantifying drug-induced changes, disease-induced changes, and it could therefore potentially add value to non-clinical efficacy testing, but also safety pharmacology. So I'll quickly move on to the next one, which is the clinical case study looking at COPD. So from this, we had 12 patients, of which 10 had usable data. These are patients who went into hospital with an exacerbation. They were then deemed clinically fit for discharge. They went home. And then every day, a doctor came to their house, put a nasal prongs on their nose, and monitored their pressure for 7 minutes every day for 30 days. And during that 30 days, 4 of the patients re-exacerbated and either came back into hospital or took a rescue pack, whilst 6 of the patients remained at home. So we wanted to see, could the attractor have kind of discriminated between those groups? So these are 2 sets of representative attractors from 2 of the stable patients. So you can see, again, it's like a helicopter shape, it's 3-armed, and it remains like that for the entire 30-day monitoring period. If you look at the patients who re-exacerbated, from the point of discharge, when they were deemed clinically fit for discharge, you can see there's a hole in the middle of the attractor. It's different to the stable ones. And that hole gets bigger and bigger and bigger and wider and wider and wider until eventually they came back into hospital. So if you quantify the standard metrics, so a nasal prong, you can only really get respiratory rate. And we know respiratory rate is a brilliant biomarker, but it's not measured particularly accurately, and it's often rounded up and rounded down. But if you do it properly, you can see that actually there's a good classification accuracy between the exacerbators in red and the non-exacerbators in blue, about 0.83, so 83% classification accuracy. However, if you use our method and look at opening on the same data, it just amplifies it and it allows you to classify much more accurately 0.96. So that's over the 30-day monitoring period. If you look at day of discharge, it's even better. So day of discharge, our method perfectly performed, small sample size I know, but perfectly performed in discriminating between those who would go on to exacerbate versus those who didn't exacerbate. And again, slightly better than respiratory rate. So again, we need to now do a prospective validation to work out, does this improve fitness to discharge decision making? So could we have changed the decision about whether they go home or not? And also, could it provide an earlier alert for patients to come into hospital? Because it's known that they often leave it too late. And if you leave it too late, that can actually impede outcome. So to summarize, the problem we have is that everyone's collecting this data, the machines are collecting this data, it's in there, but it's underused. And the second problem with conventional metrics is that baseline wonder is a problem, and it's always going to be a problem. This is a maths in medicine solution, 2 completely different disciplines allowed to sort of come together. And it's a new way of visualizing the waveform data. It uses every single data point, so there's no inadvertent bias introduction. And it gives you new metrics, which are morphology and variability metrics. What we've seen in this particular study is that sort of opening of the attractor is associated with increased symmetry of the waveform. And that suggests that there's increased work of breathing. So it's not telling you that you've been given the drug bleomycin, but it's telling you that the work of breathing is changing. And it appears to be more sensitive to change compared to conventional metrics. And so ultimately, we'd like to combine this with conventional metrics and apply it in lots of areas, but particularly in this context and to this audience, potentially detecting drug and disease effects. So I'll end there by thanking, this is just a handful of the people who were involved, but particularly Miguel Serna Pascual, who's my PhD student, who's been absolutely brilliant in kind of developing an app to analyze this data, and all of the clinical colleagues and industry colleagues who've been so kind and given us their data, and thank you for your attention.
I can't, okay, now you can hear me. I can't do anything about it. I just can speak up. So just to repeat for all of you, I said it was a great presentation, and I was actually quite happy that she left out all the math. So are there any questions? There is a question in the back, thanks.
Hi, Manasi. Thank you so much. It was a really cool presentation and an interesting way to visualize the data. My name is Charlie Dean from Cambridge Consultants. I just had a question. Your the 3 points on the waveform, given that they're a function of the frequency of the waveform, is there like a feedback loop that adjusts the period between those points?
Yeah, absolutely. So that's a really good question. So yeah, which I didn't sort of point out. So the system auto-adjusts. So you have a window of data. Let's say you've got 20 minutes of recording. And to say every 10 seconds, you say, what's the average cycle length? It works out the average cycle length. The 3 points are then a third of that. It then shifts to the next window, and then it slightly auto-adjusts. And what that does is it retains the symmetry of the attractor. Otherwise, the attractor would collapse. So that's why it's called symmetric projection. But yeah, I kind of missed it out.
Brilliant, thank you.
Thank you.
There is another question.
Can you give a feel for some of the other applications that this might kind of apply to?
Yeah, so we've applied it in lots of areas. I'm just very slow at publishing it. So we've looked in PPG signals, so photoplethysmography signals, whether we can extract more from those, blood pressure signals, looking at early infection, because we think we've got biomarker of inflammation. So it can work on, and it can work in non-physiological data as well. It can work in engineering data. That's just not my space. But yeah, just looking at a change in a sort of functional system. So yeah, it's kind of endless list, which is nice and complex at the same time.
Another question?
So Martin Oliver from Pulmosant. Anybody who's worked in clinical trials for pulmonary diseases is forced to use FEV1 as a primary endpoint. And the frustration around that is the variability seen in the recording and the interpretation of the results. Do you see this as a way of addressing that issue?
Yeah, so this is a really interesting question. And I'm not a respiratory sort of expert by background, but I'm working a lot with respiratory physiologists, and I also know how challenging it is to do an FEV1. We're really interested actually in tidal breathing. So can you tell subtle differences from actually resting tidal breathing from someone who's got pathology rather than forcing a system? But if you just did a spirometry and got a single wave, it wouldn't work there. But in theory, if you've got lung disease, perhaps your waveform will change its shape.
Manasi, Ben, can I ask here?
Sorry.
Since you're taking, their measurements are routinely made. So to progress your next steps in the respiratory field, do you need to go out and design studies to look at particular, aspects of, recovery, disease, etc., effective treatment, or actually are there data sets already out there that you can access? And actually, there's a whole plethora of data that's just waiting for you.
Yeah, it's a good question. And I think so in the preclinical space, certainly with the GLP industry data, it'll be sitting on databases everywhere. So that's there. I think in the clinical setting, I don't always know the fidelity to which they collect the data, and also the timestamps and annotations are not always perfect, because the study's not designed to do this, it's designed to do something else. So the one issue we've had with clinical data is we need to know times at which, for example, a clinical fitness to discharge rather than a diagnosis was made. We need to know when was their decision triggered, because that's the point at which we need to get. But yes, certainly with preclinical, I know it's there because we've spoken to people who have it. And I think from an industry perspective, I guess people are interested in then further characterizing, saying, well, can we tell differences between types of drug or types of disease, and are there specific signatures for that? So yeah, till we have the data. Thanks.
There is another question in the back.
Hi, Chris Rowe from Quotient Sciences. On your slide with the COPD patients, I noticed that the opening of the SPA for the hospitalized patient seemed to be more of a change from the helicopter shape to the sort of circle, and the one that said treated seemed to be just a more fuzzy helicopter shape. Do you think there's a level of detail in
Yeah, I think fuzziness is inflammation. And I think fuzziness is often inflammation in this. And I think what I'm thinking is that you get a hypervariability when you have an inflammation that is not sort of life-threatening, and you get hypovariability, you become incredibly periodic when, and that's known in sort of blood pressure with sepsis, for example, that you become, you beat like, your heart beats like a metronome. So I think the degree of fuzziness is telling us about inflammation, and we would therefore be interested in looking at drug responses pre and post challenge, or things like for sleep apnea patients, looking at the impact of the CPAP therapy, how does it change the waveform? So yeah, there's stuff in there, we just need to figure it out.
And there's one last question.
Yeah, hello, Justin Lacombe from Experic. I'm just wondering, I mean, considering this is kind of a signal, a signal analysis application for this, have you considered other sort of measurements along that line, like fractional dimension of the attractor itself, or done fast Fourier analysis on the signal?
Yeah, so well, fast Fourier analysis is kind of done already on things like blood pressure signals. We have looked at fractal, so looking at it in 3D and then saying, do you have something, don't you have something? At the moment, we're focusing on this 2D because it's kind of giving us something new above and beyond. But yeah, you could automatically then move to fractals in 3 dimensions. Yeah, thank you.
So thank you again very much for this very interesting and stimulating talk. Thanks.